Thinking that I bit off a little bit more than I could chew, trying to cover all three of these diseases in one 30-minute presentation. So I'm going to jump right into it. Before I do, two very big thank yous. One to Dr. Andy Bowman with the College of Veterinary Medicine here at OSU. Provided a lot of the data that I'm using in the first part of this presentation. And then to our Division of Wildlife staff. The deer program, they do a lot we ask a lot of our staff, and they come through in every situation to get us the information that we're looking for, and they're the reason that we have this data to present today. So without further ado, we'll get right into it. I'm going to treat this as basically three mini presentations in one, where we talk about each one of these diseases. So number one, SARS-CoV-2 in white-tailed deer. So as if deer hadn't already done enough to make the news for all the wrong reasons, they decided to become a potential reservoir host for one of the most significant zoonotic diseases that we've seen. And if you follow the news, you've probably seen some of these headlines where they note the significance of finding this virus in the deer population. So summer of 2021, there was a kind of a rash of papers that came out to describe the fact that deer can in fact be infected with SARS-CoV-2 and that they can transmit that virus to other deer. This particular one, the last one here, was from deer collected in Northeast Ohio, which is, that's pretty cool that this state was one of the first to kind of find the disease in deer and kind of on the forefront of this research. So that particular study, winter of 2020, deer were collected in Northeast Ohio. 36% of them were positive for SARS-CoV-2. Fairly significant number of deer. The data that they collected included some genetic analysis of the virus variants and of course geographic location of the deer, and it suggested that there were multiple human to deer spillover events that occurred, and that the clustering of the positives that they found along with the number of deer that were positive for the disease suggests that the transmission was occurring from deer to deer as well. I guess one important note here is that through all of this, where there's not really been any evidence that infected deer show any clinical signs of disease, but they are obviously capable of being infected and shedding virus. So given that information, we'll go into the next full year of surveillance, 21-22 season. And this was a statewide effort. Again, kudos to our staff for tackling the challenge just like they always do. And we were able to collect over 1,500 swabs from deer in 83 of our 88 counties. And we detected infection in about 11% of the deer that were sampled. So this is one of the more interesting parts of this particular part of the talk. The graph that you see there is the number of infections, COVID-19 infections in humans from January 21 to January 22. So you'll note that the timing of our sampling in this particular year was November through January, the deer season. So the genome sequencing was done on 80 of the 160 or so positive deer, and what they determined was that 71, the vast majority of those virus strains were of Delta variant, which makes sense given the timing of the sampling, where the most dominant strain in humans at that time was Delta. But the really interesting part of this is that they detected alpha variant in 9 of the samples. And so when you look at the timing of our sampling, and then you look at the timing of when alpha was present, most dominant in humans, that particular strain had died out in humans almost 5-6 months previous to our sample collection in deer. So it's fairly significant evidence that the deer are able to maintain the virus in the population all by themselves. All right, moving on to 22-23, this past season, again, over 1,500 samples collected across the state. However, this time only detected SARS of 9 deer, so about 0.5%. Why is it so low? Well, you think about what's been happening as of late. there's decreased SARS-CoV-2 in humans. The current variant, Omicron, the most dominant variant at the time, may not necessarily infect deer as readily as the other strains. And there's a possibility that we might have some increased deer immunity. So those are all hypotheses for that, but of course this is hot off the press, so not a whole heck of a lot of time to kind of really dig in. But that's where we stand right now. So summary of SARS-CoV-2, we know that human to deer transmission does occur. Occurs across the state. Urban areas, rural areas. We collected one deer in 21 from Lawrence County, one of the more rural parts of the state. And that one deer that we collected was positive for SARS-CoV-2. So it is across the state. We do not know, this is the number one question, how is it getting from humans to deer? We don't know, is the only answer I have for you. There are hypotheses, but at this time we don't know how it does occur. We know that the virus is transmitted from deer to deer, and this is most evident with the persistence of that alpha strain in the second year of surveillance that persisted in the deer population many months after it already died out in humans. And then one thing that we have determined, don't ask me how they determined this because I'm not an expert in that field, but that the evolution of the virus itself is occurring at 2 to 3 times the rate in deer as it does in humans. There's a higher rate of mutations in the virus strains, and that these are mutations that are rarely seen in human strains. So given all of these factors together, kind of the one big question remains, the most important part of this, is will deer become a permanent reservoir for SARS-CoV-2, and could new strains develop in the deer population that could ultimately spill back to humans? So we don't know the answer to that, obviously, but it does give reason to kind of keep an eye on this and continue with some level of surveillance of the virus in deer as we move forward. All right, that's SARS-CoV-2. Let's talk about hemorrhagic disease. So hemorrhagic disease, you hear it called EHD. Here, blue tongue is sometimes referred to, but in Ohio, the specific virus that we find is the EHD virus that causes hemorrhagic disease. And just a quick once over here, hemorrhagic disease is a virus that's transmitted through the bite of a culicoides vector. This is common names for that would be a gnat, no-see-um, midge. This is a very seasonal disease. We only see this disease occur in Ohio in late summer, early fall. The first frost of the year in the winter will kill the vector, and hence transmission of the disease will stop at that point. So it's a very seasonal disease. Long-term implications for deer are almost none. However, short-term implications are that we can have significant localized mortality in a short period of time. If infected in Ohio, the disease is, on an evolutionary scale, relatively new to deer in Ohio. And if infected, few of the deer will survive, if any, actually. And they will mostly, once the onset of symptoms occurs, about 7 days after infection, they will die within about 72 hours. They die as a result of internal hemorrhaging, so they basically bleed out from the inside. They have extreme fever with the disease, and that is one reason why you often find them near water when they die, is they're seeking, try to cool themselves off. Also, they become extremely dehydrated because of the hemorrhaging that's occurring. And then one final kind of symptom that you might see in that 3-day window, if you're lucky enough to, or unlucky enough to see one, is abnormal behavior, the little fear of humans, they act disoriented. And this has been a particular issue this past year when we had EHD in areas with lots of humans, because we had some deer that were seen by humans that weren't acting quite right. And for whatever reason, it's unknown at this point why, but they got termed zombies. Now, I'm somewhat of a science fiction fan, and my definition of a zombie is either it eats brains or it rises from the dead. Neither of which is true for these deer that are infected with EHD. So where this term came from, not so sure, but it's catchy and maybe it gets clicks. So we get these rash of news clips about zombie-like deer being found and seen in Ohio. Unfortunately, some of these headlines, I talked to the reporter and asked them kindly, please do not refer to these deer as zombies or call it zombie deer disease, but obviously with the headlines you see, we're not listening. It made it all the way to the New York Post, unfortunately, about zombie deer. And this one was particularly troubling. They actually saw zombie deer in parts of Ohio and Indiana, but not only did they see them, and in the article, of course, their descriptions match Epizootic Hemorrhagic Disease. But the biggest issue here is that they have this title up here, zombie deer. What do you need to know? Well, zombie deer is another name for chronic wasting disease. First of all, no, it's not. Second of all, you've got two different diseases that you're talking about in the same news article, which causes confusion among the public, obviously. So that caused us some issues this past year, but we worked through them, as we usually do. So with hemorrhagic disease, we typically experience some every year. And we get reports from the public, from our field staff, and those are tallied in our online database system. And then anytime we get reports of dead or sick deer, we do try to collect them during this period of time, late summer, early fall, to try to confirm with laboratory testing, confirm the presence of EHD. And so if you look over the last several years, 2017 was a fairly big year for EHD. We had over almost 1,400 cases reported to us and confirmed the disease present in 17 counties. But our more typical years are those that follow, where a handful of counties will confirm the disease in, and we'll have 100 or maybe 200, 300 reports of disease from the public. But this past year, 2022, we had over 1,500 reports of dead or sick deer, and 47 counties where we were able to confirm presence of the disease. That is far and away the largest geographical extent of EHD we've ever found in a single year in the state. It was a very odd year for EHD. Not only the sheer number of counties that were positive, but the way that they were distributed, the way that the reports of sick deer were not distributed, for instance. So we have some obvious areas of the state where EHD hit particularly hard, Southwest Ohio and Southeast Ohio. But we also have a large portion of the state, particularly North Central, thank you, very, very much appreciated, where we confirmed the disease in the one or two deer that we got calls about and were able to collect and get to the lab. But other than that, there really weren't a whole lot of reports coming in of dead or sick deer. So an odd year in that we were able to confirm it in a whole lot of places, but we did not, a large portion of those places where it was confirmed, we did not see a whole heck of a lot of significant mortality. All right, so what do we do about hemorrhagic disease? Well, first off, there's nothing preemptive that we're going to do about hemorrhagic disease. It's going to come and go, as it has in the past and will continue to do, and when it comes, it's going to kill deer. But again, it's a localized thing, and deer populations recover. So the one thing that we can do is we can react to it if the circumstances deem it necessary. And the main way that we can react to it is through regulatory means. We can reduce antlerless harvest in those areas to kind of give the deer herd a break and allow them to recover. And the most recent example of that was in 2017, the last major outbreak of disease before this year. And this was highly concentrated disease in eastern Ohio, and particularly Jefferson County. So much so that we decided to reduce the bag limit in that particular county to give the deer herd a little bit of a break, given the heavy losses that they experienced due to hemorrhagic disease. And so how that worked, I mean, here's the chart, population trend given by our annual buck harvest. And you'll see in 2017, buck harvest took a huge dip. There wasn't many bucks out there to harvest because they had all been killed by EHD. But we reduced the bag limit, gave the deer herd a break, and it did just what deer herds do in eastern United States, and that is grow. And so while HD does kill a lot of deer, it causes concern among the public to a great degree in some cases. It is certainly not a long-term threat to the sustainability of deer populations. All right, let's move on. I've got 10 minutes to cover chronic wasting disease. That is a tall task. So it's going to go a little quick, but hopefully I can get through. I assume most people in this room have heard of chronic wasting disease, but really, really quickly, it is an always fatal neurological disease that affects members of the deer family. It is caused by a misfolded prion protein. So this is not a virus, it's not a bacteria, it's not something that's living. It is a protein, a string of amino acids. And the protein becomes misfolded, and whatever mechanism is involved there, it initiates a chain reaction that causes the other prion proteins to misfold, creating holes in tissue, particularly nervous system tissue, most notably the brain, and you do not do well with holes in your brain, so you die. It's spread through animal-animal contact and through the environment. So infectious prions can be shed from deer through saliva, blood, feces, urine, and through decaying carcasses, released into the environment where they can remain infectious for years. So you may not have any deer on the landscape that are positive for disease, but if they come and pick up some soil somehow or eat a plant that has had prion uptake, you might have infection, even without another deer to infect them. There is no cure for infected animals, and the CDC, while no strong evidence exists that this is a human disease, they do recommend that hunters do not eat the meat of deer that test positive. So that's CWD in a nutshell. What we've seen over the last several decades, prior to 2000, this was the known distribution of CWD in North America. Since that time, so it was first documented in a research facility in Colorado in the 60s. But up to 2000, this is all we knew about chronic wasting disease. Since then, we have seen spread far and wide, and as you up through 2010, 2015, 2022. So you see a pretty massive spread of disease now confirmed in 30 states in the United States and several handful of provinces in Canada. Don't want to do that. Okay. And you'll notice a couple of spots there in Ohio. And so that's what we'll talk about real quick. Ohio's first free-ranging detection of CWD was in Wyandot County in the fall of 2020. This is a hunter-harvested mature buck that was sampled just as part of routine surveillance by a taxidermist. That is the actual buck there on the screen. And this was out of left field, honestly. Wyandot County was one of the counties we had deemed very low risk for the introduction of chronic wasting disease based on the suite of factors that we look at. So how did it get here? We get asked that a whole lot. How did it get here? Where did it come from? Well, short answer, we didn't know until I started to put together this presentation. And I don't know if you've seen it yet, but I think I figured out where this CWD deer came from. It was imported from Canada. I'm glad to hear laughs. That is a joke if you didn't get it. We don't know where it came from. But it's here, and it doesn't matter where it came from. It's here, and we have to manage it the best we can. So we find the first deer is positive. We initiate several controlled hunts throughout the rest of the deer season to try to increase harvest in the area. We were lucky that we were close to Killdeer Refuge, where we had access to our own property to let people on and shoot some deer and get a handle on what are we looking at here? What's the geographic spread look like? What's the number of infected animals look like? So after that first year, we got one additional positive doe on the refuge. All right. So prior to the next deer season, initiated a disease surveillance area. Parts of this, mandatory sampling during portions of the deer season. Hunters are required to bring us their deer for sampling. It prohibits baiting and feeding, and their carcass movement restrictions, because we don't want folks coming to this area, killing deer and taking it elsewhere without certain other rules that are in place. So the results of our first full season of surveillance in that zone, we were gifted 9 additional positives. Thankfully, they were all within the same geographic area, relatively speaking. So that was the good news. And given the results there, made some adjustments moving forward to try to further manage the disease in the ways that we could. So we expanded the disease surveillance area to include the entirety of all 3 of the counties. And the primary reason for that was we wanted to introduce some new hunting regulations to combat chronic wasting disease. And our hunting regulations are promulgated at the county level, so it only made sense that we include the entire county. So the primary changes here were an earlier start date to our archery season in those counties, and an early firearms season. In this case, a 3-day firearms season. And the idea here is to try to harvest deer earlier in the season, as early as possible, really, in order to combat the risks that are associated with the breeding season. Deer do lots of contact with one another during the breeding season, as you might imagine, and they move long distances. So we wanted to try to remove as many positive deer as possible prior to kind of the annual shuffle, if you will. So how did that work, the impact of the early gun season? Again, the main goal was to shift the harvest earlier in the season. And so here you see the cumulative harvest. This is the 3-year average, so 19, 20, and 21. This is prior to the regulation being in place. You see that the cumulative deer harvest across the disease surveillance area as it rises through the season. So obviously the big spike there is our traditional 7-day gun season, right? And so what we would hope to do is to shift that curve to the left so that we kill these deer earlier. And, I went backwards. And we did, actually quite effectively. We killed almost 700 deer in that 3-day season across the disease surveillance area, which pushed us to that 1,000 deer mark across the area, which is about 25% of what would be the total season's harvest. So we reached that level of harvest almost a month earlier than we would in traditional circumstances. So in that way, it was a successful move in that we did kill more deer earlier in the season prior to breeding. But as you see, the kind of the curve at the top there, we didn't really increase the number of deer harvested by any it was fairly negligible amount. So harvest earlier, good. Kill more deer, not really. But the results of this year's surveillance, we had 7 additional positives. Not quite as many as the year before, but you look for disease where you know disease is present, you're going to find it. Essentially, is what's happening. We did add a new township to the map here with Grand Prairie over here in northern Marion County. And we see some eastward movement there as far as where the positives are occurring. Most of the positives this year were still within that really kind of tight core area, but we are seeing some, a little bit of movement outward. All right, final thoughts. It's not going to go away. This is, given the number of positives that are on the landscape, there's likely enough environmental contamination that will never see the end of CWD. But it is currently at very low levels. And we can hope to maintain the support that we have to continue the management activities that we're doing that will slow the spread of the disease and maintain that low infection rate. A couple of these are the earlier harvest, more harvest, postseason removals that we have conducted to try to remove positives in areas where we know disease exists. And the success of our CWD management efforts will depend, not entirely, but almost entirely, on hunter cooperation, their acceptance of our management actions, their compliance with our rules, particularly our carcass movement rules, and their proper disposal of deer parts. And the reason that is so key is that there are lots of deer carcasses moving long distances. And pretty much that happens pretty much everywhere in the state, but if you look at where the disease is currently, which is right around Killdeer Plains Wildlife Area in Wyandot County, Wyandot and Marion County, these are the home addresses of folks that harvested deer on public land in Wyandot County, which really the only major public land in Wyandot County is Killdeer Plains. So these are people that are harvesting deer in an area known to have chronic wasting disease. You see the distribution there. The vast majority of the deer that these folks are harvesting were taken on Killdeer Plains, where roughly almost 1 out of 100 deer currently are positive for chronic wasting disease. We know from survey data that half of these deer are likely processed by the hunter themselves. And unless the hunter takes it upon themselves to properly dispose of the parts, wherever he deposits those parts could serve as a source of disease introduction. So, you see the red circle there. Not saying, just saying. Questions? I'm sorry, you get in the mic, I couldn't hear you.
Yeah, I was just curious if you employ, and if so, if you find success with locating and treating carcasses alongside your surveillance, so burning or burying?
No, given kind of the distribution and the other, knowing that we're not going to